Hey guys, welcome. So remember, if you have questions, just type them and then I will answer them as quickly as I can. And this is Katniss. Get out of there. Okay, Tony, I see your request for 4.9, number 17. Let me pull my copy of that up. Okay, so number 17 asks, are homologous chromosomes identical? So homologous chromosomes are not identical. The only things that are identical are sister chromatids. Homologous chromosomes, however, code for similar things. So for example, while they might have the same types of genes, like homologous pairs will both talk about the same types of genes, but they will not have the same alleles. So that means, say you have homologous pairs, remember, come from a maternal and a paternal side. So your maternal chromosome might say green eyes and red hair. Your paternal one will also talk about eyes and hair, but it might say blue eyes and brown hair. Whereas a sister chromatid would say the exact same thing. So if you had a copy of that chromosome, both would say the exact same thing. So homologous pairs are not identical, but they're talking about the same things, whereas sister chromatids are identical. Does that answer your question, Tony? Anybody else have any questions? Okay, number 33. So 33 asks, what does the word aneuploidy mean? And so aneuploidy is just referring to chromosomes that are of an abnormal number. So for example, if you are monosomic or trisomic, so having 3 chromosomes instead of just 2 in a certain number. So those who have aneuploidy usually go through non-disjunction. So they have the wrong number of chromosomes or an abnormal number of chromosomes, which is what the word aneuploidy means. So, for example, those with Down syndrome have aneuploidy. Anybody else? Tony, let me know if you need any more clarification on number 33 or if that was good. Okay. Okay. So number 29 asks what do scientists use to stain the chromosomes to make their distinct structures more visible? And that's the Giesma dye, or the G staining, or process of G banding. So Giesma dye is, which is spelled Geisma, I believe. And it's a dye that adheres to the proteins that are in DNA, specifically between the bonds of adenine and thymine, to darken those areas more substantially, creating a banding pattern. And then, of course, we organize homologous pairs by chromosomes that match in banding pattern, size, and the placement of the centromere. Come on. Any other questions, or are you guys just going to watch me pet my cat? Because that's not creepy at all. All right, anybody have any other questions? Okay, 4.8, number 8, last section. So would this mutation affect every cell in an individual or the entire body? Okay, so for germline mutations, it's going to affect every cell, because remember, those originate from your gametes. So, for example, if your parents were to have mutations within their gametes when you were being developed, then every cell that comes from those gametes is going to have that mutation present. So germline mutations, remember, deal with the gametes, which when they combine form a person. So if the gamete's infected, then all the cells that come from those gametes, which will be all the individual cells, will be infected. Somatic mutations are just in your body, other body cells. They don't get transmitted to or hereditarily. Sorry, there's a cat fight for a second. But they do not get transferred to progeny. They just affect one region of the body. So a somatic mutation is only going to affect the originates. So for example, if you have skin cancer, that's not going to get passed down to anybody, any children you may have, but it's just going to affect the one area that you have on the body. So short answer, germline mutations, yes. Somatic mutations, no. Or some germline mutations will affect the entire body. Somatic mutations will only affect an individual certain area. Was that all you wanted to go over, Anthony, or did you want to go over any of the other sections, Tony? Okay, number 9 on 4.8. Okay, so chromosomal alterations is just all the other types of mutations that aren't substitution/point mutations. So when it talks about chromosomal alterations, think translocation, think insertion and deletion, aka frame shift mutations, and think inverse mutations. Those are what they're going to be talking about. So what are the subcategories of chromosomal alterations? That is translocation, frame shifting, so insertion or deletion, and inversion. So those are the three. It can change multiple DNA bases, or it changes multiple DNA bases. With insertion and deletion, it can delete or insert only one, but it will affect multiple RNA codons. So the answer for chromosomal alterations for DNA bases, 1 to more than 1, and RNA codons, more than 1. It will always change the amino acid sequence, and usually more than one amino acid is changed as a result of those. Whereas with point mutation, it is just silence, missense, nonsense. It only affects one DNA base. It can only affect one RNA codon. It does not always change the amino acid sequence, because remember, silence does not change it. And usually only one amino acid can be changed. Just one. Just think of it that way. They're changing one, and sometimes it's a stop. Hello, odd. Here's the other one. They don't actually love me this much. They just want to get fed. So you're impeding on their dinner time right now. You're welcome, Tony. Okay, come on. Come on, Katniss. have any questions, just ask them and I will stop practicing guitar and answer them. So. Again, guys, just ask a question or just ask me to explain a topic and I will do that. Okay, again, the way this works is if you just ask me a question or ask me to review a topic, and I will walk through it. The way you ask a question is by typing in the chat bar. Okay, 5-minute warning. If I don't have a question in the next 5 minutes, I'm just going to assume you're lurking for whatever reason. You're welcome, Theodore.